Good afternoon, everybody. It's a pleasure to be here, to be back to the Max One Summer School. Thanks to Jürgen for inviting me and for giving me the possibility to present my results. I will also try to justify why it makes sense for me to be here in this context. But I would like to start saying that I'm a proud attendee of the 4th edition of the Max One Summer School that took place here in Munich in 2012, so a while ago. And there was also pictures of me acting as a guinea pig of a magician doing a show during the school. And I work at the University of Copenhagen, what now is Globe Institute. And the Globe Institute is a highly interdisciplinary research unit that focuses on addressing basic scientific questions. So we are spread in multiple venues in Copenhagen city center, and part of us work in the former city hospital and at the Geology Museum. And certainly, some of the sections at the Globe are well known for their activity on ancient DNA and genomic analysis. Tom Gilbert, Eske Willerslev, the founders and the pioneers in the field. Many first and high impact papers, including the first ancient human genome. That was in more than 10 years ago. But at the same time, we also investigate another category of ancient biomolecules, that is proteins. And when I arrived, as Carlo mentioned, in 2009, there was this strong center emerging in the University of Copenhagen panorama, but also another very important center. So geogenetics, where I work, is strong for ancient DNA, was strong already for ancient DNA at the time, but few meters away, a few hundred meters away, 5 minutes by bike at the Panum Institute, there was another center that was emerging, super strong in biochemistry and proteomics. And this is how Nature in 2007 announced the opening of this center that unfortunately is going to close in the near future. And that year, I met Jesper Olsen, one of the leaders in proteomics. And basically, my selling point was geogenetics is very strong in ancient DNA. CPR is very strong in proteomics, modern proteomics. Why don't we try bridging working on ancient proteins? And that's how we started. So, but why should we bother about and working on ancient proteins? What's the message? What's the relevance? How does it fit in this context? I modified my slides yesterday, well, this morning, basically. And I found a reference that came across it for a comment paper we are writing. Also, the reference is old. It dates back to 65. And last author, Linus Pauling, so not the last one, twice Nobel Prize, once for chemistry and one for peace. One of the few with single, with a non-shared Nobel, with two, the only one with two non-shared Nobel Prizes. And it was the report of a conference, basically. And we go through the first two paragraphs very quickly. And basically, there were these exponents of chemical paleogenetics that were seen, that were under crossfire from both a community of evolutionary and taxonomists, evolutionary biology and taxonomy, and another community of pure biochemistry, very unorganismal. And basically, the argument of the biochemist is that characterization should be focused on relation, that the characterization of relations between organisms should be a second-rate interest for biochemistry. And they were saying that people should focus on molecular mechanism underlying the observation of classic biology. And it is true that the concept of mechanism, molecular mechanisms should be crucial, but at the same point, it comes out that these molecular informational macromolecules, very interesting term, DNA, RNA proteins, also called semantids, as a unique role in determining the properties of living matter at three levels, at three different time scales. So there are the short time biochemical reactions, and this was perfectly described yesterday by the other keynote lecture, but there are also medium time ontogenic events, so disease, physiological processes, aspects associated like pandemics, so stuff that happens in a human time frame. And then there's long-term evolutionary events. And the conclusion is, at the paragraph, if one loses sight of the slower processes, one also loses the links between the component of fastest processes. So this is a possible way to justify, explain, or contextualize why it makes sense to also investigate ancient proteins. Also in a place like CPR that is hardcore focused on molecular mechanisms and fast biochemical reaction. And it's interesting to observe how consideration done almost 16 years ago are absolutely valid today. What happened in the meantime is that there was another one of these macromolecules, informative macromolecules that took over, that is ancient DNA. Here in this review, updated at 18, together with Jürgen and other co-authors, we describe what was the status of the art at the time. And it's not a coincidence that the first parts of ancient protein analysis actually starts before ancient DNA analysis in 1954 with strong support in the US from the Carnegie Institution for the identification of amino acids already at the beginning of the second half of the last century. To cut the story short, we were saying in the meantime, ancient DNA took over. It was even worth a Nobel Prize awarded to a colleague in another Max Planck institution, the one in Leipzig. But so where ancient proteins can have an advantage, and basically the key is here in the subtitle. So where DNA degrades, protein might persist, and this is dated at June 19. And actually, that's the first line of the subtitle should be slightly modified because might should be removed with do persist. We now know that we can retrieve proteins and accordingly informative macromolecules, genetic information farther back in time than DNA. And that's the best option when DNA is not available. So this was made possible by the introduction since 2000 of mass spectrometry applied to ancient proteins and then the development of all the proteomic approach. So to answer the question you asked yesterday, there was actually a totally unplanned but perfect assist about what happens when we die. This is how ancient proteins look after we die, and or how they look when we can retrieve them after millions of years, up to millions of years. They are very damaged, contaminated, and available in very limited amounts. So basically, doing ancient proteins in a way is asking for troubles because many factors are complicated, and we'll see why. So contaminated because, of course, staying in the ground for geological times, there's integration of contaminants, bacteria, microorganisms, and leach of the original proteins. Sometimes it's on purpose that there's contamination because, for example, in painting material that have protein traces, this material is deliberately mixed with many other, for example, in a paint layer with the dye that is a mineral that gives the color, and it's available in very limited amount due to degradation or again, by design in some sampling experiments, because when you look and work with artistic material, you cannot have samples the size of a stamp, but the curators are only able to give you very tiny amounts. So The sample preparation is very simplified, and the intention is to throw as less as possible because everything is precious. This is one of my last appearances in the lab. Biomineralized samples are collected, and with very high-tech biotech, they are fragmented, powdered, and extracted, usually demineralizing using acid or ADTA. And then very simple workflow. So guanidine hydrochloride, reduction, alkylation, and digestion with final immobilization on stage tip. And then LCM, SMS, and most of the cases, but not always, analysis with MaxQuant. Let's have a look at some application, and we start with the most recent material, and we progressively move farther back in time. So application with artistic and archaeological material. A few years ago, again, with colleagues at CPR, we had the possibility to analyze a layer that was covering a fresco painting that now it's at the National Gallery in London. And it was interesting, not particularly exciting, but what we found was collagen, so the use of animal glue, but together also proteins from chicken and duck eggs. This was interesting, but the most interesting part was associated with molecular characterization of the molecular damage. So measure of the amidation, super common. And it's possible to see how the fact that the collagen and the glue is boiled immediately increase the amount of the amidation. But most of all, and this was based on the use of the dependent peptides function that is present into MaxQuant, it was possible to characterize let's say a blind search of PTMs, and both Diane and Patrick that were involved in the project identified a couple of modifications that were particularly interesting. One associated with photooxidation, so exposure to light, not a surprise, and interaction with starch with advanced glycation end products. So why can protein be useful, or protein analysis can be useful here? First of all, to reconstruct the technology, the technique of production of the artistic material or the preservation, and then to study its degradation. This was part of a training network that I coordinated between 16 and 19 that also involved Jürgen, and that's how we started working together, and other groups in around Europe. Another application associated with something similar is on archaeological material. So identification of the species of ivory and bone museum objects using minimally destructive approach. So there's a collection from the Metropolitan Museum in New York of objects that are made in bone, and this approach is focused on identifying from which species these objects are made. Also in this case, for reconstruction of natural resources exploitation, for example, and of course for characterization of the history of the object. Let's see another application of material that is more or less in the hundreds of years range. And it was interesting to hear the presentation yesterday about metaproteomics, because probably this is the only quantitative metaproteomic approach on ancient samples published so far. And we use a material that is not particularly noble, but very rich of proteins because it's dental calculus, so mineralized dental plaque that accumulates on teeth when there's not good professional hygiene. So this is material from Denmark, is about 1,000 years old, and you can see how rich is the sample in a way. And the calculus basically was scraped off the teeth and demineralized in acetic acid. Also in this case, lysis in guanidine hydrochloride and was labeled with TMT and analyzed. So here is the description of the workflow. There was single shot analysis and then merging and TMT labeling and analysis. what we see is that the population of these individuals from this cemetery basically are divided into groups. We cannot talk about healthy and diseased, but certainly two groups with different profiles oriented toward more or less pathological status, and the comparison with modern plaque and calculus, as you can see here, clearly shows that the profiles of the two ancient groups are different. Groups, so species associated with what is called the red complex in one case, and less problematic species. But let's go again farther back in time and now focus on paleontological and paleoanthropological applications. So we were saying ancient DNA, we already revealed the end of the story. Ancient proteins last for longer, but if we have a scale of 2 million years, what we can see is that most of the ancient DNA studies focus on the last 10,000, 100,000, if we stretch it, 200,000 years. The most ancient DNA identified so far from an organism, so from fossil material, is a bit less than 1.5 million years old, but it comes from a super cold place from Siberia where biomolecular preservation is higher. On the contrary, the most ancient DNA from a temperate environment comes from Spain, and in particular from a site we will see again, Sierra de Atapuerca, north of Madrid, just outside Burgos. And it's a bit more than 400,000 years old, and it's material from a species called Homo heidelbergensis. And in this range, there's almost nothing. So paleogenetics is not particularly effective, with one exception that I will mention immediately. But even if we start to work with paleoproteomics, we see that we can retrieve some sequences, not a lot, you will see it immediately. We are happy when we cover 1,000 amino acids, but still better than nothing. And there are precious information that can be retrieved from that context. So ultimately, paleoproteomics enables molecular-based phylogenetic reconstruction beyond the limits of ancient DNA preservation. This is the exception I was saying, 2 million years old DNA for the farthest north of Greenland. So extreme environment, 2 million years old, they say could be even 2.5. But this is extremely interesting because if you go that far back in time, you start to be in an epoch where the climatic conditions were very similar to ours in terms of high concentration of CO2 in the atmosphere and high temperature. So maybe there could be genes of interest for adaptation to high temperatures. But with proteins, people started to characterize dinosaur samples sporadically, I would say, and it was only based on retrieval of collagen sequences. And this was not particularly interesting because collagen is not particularly variable. It's a boron molecule, a glycine every 3 residues. So just changing slightly the number of peptides from the same sample involved in the phylogenetic analysis, you basically end up with placement of dinosaur next to reptiles or next to birds. So very fragile structure. We came up a few years ago with a series of papers where we showed instead the possibility to recover proteins in the million years range, focusing on a not immediately recognizable source of ancient proteins. In particular, the site we started working on is called Dmanisi, and it's in Georgia, in the Caucasus area. And it's at the confluence, as you see in the image below, of 2 rivers. So on this plateau here, there was excavating medieval stuff, but they started to find in the lower layers fossils from extinct species, and they dated this to almost 2 million years ago. This is a very important site because they found this together with the rest of the fauna material, which are the most ancient hominin, so the species that together with us are in the branch after the split from the ancestor of chimps, are the most ancient specimens of hominins outside Africa. Probably Exynthoma erectus, but there could be more than one species here, and depends who you talk to. Obviously, we had access not to that material, but to something much less attractive, and it is just bone fragments from faunal material, bone and teeth fragments from faunal material. Working on dental enamel, which is the hardest material in vertebrates, we started to see something. Also because, so the proteome is not particularly rich, not even at the source. It's less, about 10 proteins, let's say. And it's already damaged in our mouth, in our teeth, in our enamel, because in the final phase of enamel development, amelogenesis, some proteases, the two listed at the bottom of the slide, KLK4 and MMP20, are released to cut the other proteins that have a function of scaffolding for the formation of the enamel. And it's in this phase that the protein component drops and the material gets further mineralized and hard as it is in the mature form. There's one, amelogenin, that is particularly interesting because it's present in two isoforms, X and Y, coded on non-recombinant portions of the sexual chromosomes. So if you find, and they have different sequences. So if you find AML-Y specific peptides, that's a male marker. So you can call male individuals. For female individuals, we developed a quantitative method based on signal intensities to statistically confidently call female individual. There was a material that was million years old, need for a serious change in terms of sample preparation. It was easy. We went even farther because we basically skipped everything. We just added acid, demineralized, and put everything in a stage tip. So counterintuitively, this is non-digestion based bottom-up proteomics, simply because the peptides are already damaged, so the proteins are already hydrolyzed spontaneously over time. So in a way, it's we can call it shallow proteomics. Top and bottom are very close. And so demineralization, immobilization on stage tip, and straight in the instrument. We described this in a Nature Protocols paper that was released a few months ago now. And the reason why proteins persist so far back in time was characterized by our colleagues few years ago. This was 16, if I correctly remember, yes. And the reason is it's the close interaction between the mineral matrix and the proteins that, or the protein fragments that stabilize this. So they took a set of proteins, they extracted proteins from ostrich eggshells, and demineralized, in this case also digested, and they saw that basically the peptide that lasts longer from the hottest sites in Africa associated with human evolution, literally Oldupai, you see that those peptides that are better preserved and last that long are present acidic characteristics that favor the interaction with the mineral matrix. On the other hand, what I started to see when I was analyzing the Manisi-fauna material was that these peptides spontaneously generated, so there was no tryptic digestion, started to align with the sequences they were matched against, and with a particularly interesting profile, because you see these peptides. Let's take one like this one. So you have the peptide, and then the same peptide, -1 amino acid, -1, here is -2, -1, -1. So this ladder, and this is terminal hydrolysis that leads to this profile. We clearly saw that when you extract from bone, there was collagen and not much more. When we started to extract instead from dental enamel, recoveries were a bit higher. These are the numbers we're talking about, so no big excitement. But to test about the authenticity, we check for the amylation, and we see that the values compared to the control are almost at saturation, almost full and complete deamidation. With the method that we used for the fresco protective layer, we applied it to these samples as well, and we start to see that materials that have, sorry, that control materials, so medieval material, for example, have very limited amount of damage, and these are different forms of damage associated to tryptophan, very limited damage extension, while with ancient samples, this is much more advanced, and it's interesting to see that ancient samples with better preservation have a lower amount of damage than terrible ancient samples. Another aspect, terminal hydrolysis, as I mentioned before, so you have this ladder of peptides and peptide length distribution, where you see that the ancient fragments are progressively shorter and shorter. So this is also another favorable quality control. Phosphorylation, believe it or not, is there. And we see it associated with serine. And if we do the analysis of the pattern, and this is something that Jesper saw it at first sight, you see that the second trailing amino acid toward the C-terminus is very frequently a glutamic acid. And the motif, so SX, serine, whatever, glutamic, or another phosphorylated serine, is recognized by the FAM20C family, which are associated with phosphorylation of extracellular proteins, which makes sense because the target of what you're looking for is extracellular, and involved in regulation of biomineralization, so it makes further sense. We can tell the sex of the individuals, so male or females, and we will see how this can be interesting. We identify the male version, AML-Y version of, cervid and ovicaprid material. And this was the paper, because in the end, what matters is the phylogenetic reconstruction. So the point was from a rhinoceros material to reconstruct partial sequences. And here all the X's is a portion of the sequences that we were not able to reconstruct. And of course, if you just use max quant in its basic function, what you recognize is only peptides within modern variability. And what is interesting for us is detection of polymorphism and amino acidic substitution. That can be done with dependent peptides or with other tools, for example, pFind or the spider algorithm of peaks homology search. You align with modern variability, with extinct variability, back translating genomic information. And what's possible to see, for example, in the case of our rhino that was associated with a group that contained a terminal Stephanorhinus and woolly rhino. It was interesting to try it with rhinos just to see what happens, but let's go further let's go closer to us. So the next paper was characterization of this quite mysterious species, Gigantopithecus blacki. Mysterious because is supposed to be 3 meters tall, male individuals, 300-400 kilograms heavy. And all what we have is a rich collection of material from China, in particular for Chuifeng Cave in this case, a lot of teeth, but only 4 jaws. And that's all what we have. No other parts of the skull or of the postcranial skeleton. So we had a lot of teeth. We are good at extracting protein from dental enamel. We did that to answer the possible phylogenetic placement of this species. And in particular, was it associated with the Hawa branch or the branch that leads to us, hominins, farther back on that branch, or more associated with orangos? This is the area, tropical weather, so not south of China, not associated with favorable biomolecular conditions, still in a cave, so less fluctuation of temperature, a vision of the landscape and a vision of the inside of the cave, and now how the tooth looked when it was discovered. So due diligence, the amidation, very high. Peptide length distribution, also in this case comparable to what we found before, a bit shorter because the sample is a bit older, it's 1.9 million, and comes from a warmer environment. Also in this case, phosphorylation with the serine, whatever, and glutamate amino acid motif. And if we build a phylogenetic tree, we see that that species, Gigantopithecus, has as a closest living species orango, so the various Pongo species, the Pongo genus, proving that the other possible hypotheses had to be discarded. We moved a bit farther and we focused our attention on our genus, not our species, but our genus. And we had access to a fragment of dental enamel of Homo antecessor. This is the specimen from Spain I was mentioning before. This is where we went to visit the site. This was the week before of Max One Summer School Barcelona 18. Petra is the PhD fellow supervised by Jürgen here. And basically, it's a site, they have everything. Neanderthal, Sapiens, Heidelbergensis, antecessor. Here is the position of the site. This is the fragment we analyzed. I continue to look at this and I say, oh, today we could do much more, do many different things with this, but this is how science evolves. It was directly dated, so between 750, 950,000 years old. We retrieved proteins, again, made perfect sense with in terms of diamidation and peptide length distribution, phosphorylation in this case as well. It's a male individual, so we identified the equivalent of the human AML-Y sequence most frequently observed. Someone in the lab started to call this peptide Smurpies. And so we have Smurfies, we don't have Smurpies, that become ordinary jargon. Male individual, and his position, phylogenetic position, is basal to what comes after. So Denisova, Neanderthal, and Sapiens. But at the same time here, we start to see the limit. With 500 amino acids, 5 to 1,000, you cannot say much. Is it a direct ancestor? Is it on a sideline? This is the limit we are working on we are trying to overcome to basically achieve a full, much richer proteins. But if we focus on human evolution, let's look at what has been covered and characterized with ancient DNA. And we see part of our species, Denisovans, Neanderthal, Heidelbergensis, and that's it. There's material that is much older, much younger, like Floresiensis, probably 50,000, 80,000. A terminal erectus, maybe. But this comes from Indonesia, and the conditions are adverse for ancient DNA preservation, as far as we know. Homo naledi from South Africa actually is not dated at 1 million, 2 million, but is more or less same time as Floresiensis. We don't have DNA from this species. We covered antecessor as well. And now we say, yes, but if we can retrieve proteins and genetic information in the 2-3 million years old, well, the horizon broadens significantly for human evolution. And instead of covering the last 10%, 5% of the path, we almost reach 50%. And we will see some of these species. In particular, we will focus now on Paranthropus and others. This is part of the second training record I coordinated that got closed end of last month. And also in this case, here he's one of the PhD fellows, of the 14 PhD fellows, friendly nicknamed the Pushers, that were part of the project. And also of the advanced ERC grant that I'm responsible of. We have a preprint out. It's a bit old now, it's more than a year, still under review, about the characterization of the proteins from Paranthropus. This is the work of Palesa, Claire, and Ioannis as leading first authors. Material from South Africa, and particularly from a cave called Swarttrans, dated at about 2 million years. Of course, the first material that we receive is always not top quality. And in particular, you can see here how the 4 teeth are not exactly in the best shape. Belong to this species, Paranthropus robustus, one of those hominins of the robustus line, over the robust line, with chewing adaptation for advanced vertical forces, so big dentition. Multiple sides, small incisor canine, and huge molars. This is how they look a bit more in detail. And the recovery here is doing fractionation and associated with between 700 and almost 450. 2 of them are males. For 2 of them, we see one of them, we see an amino acidic substitution. We are speculating that it could be an indicator of possible 2 groups, also in connection with the morphological evidence. An heterozygous individual. The last and most advanced application, the most ancient individual here is that I want to present today, is a 20 million years old specimen from, in this case, we have to go cold, the northernmost part of Canada. And this is Ryan's work. So we're talking about the archipelago on the northern part of Canada, subpolar environment, specimen dated at almost 22 million years. Particularly rich in terms of fossil. And if we look at, for example, the set of proteins that we start to retrieve, we go a bit farther from dental enamel proteins only, because we start to see collagen and proteins associated with the most abundant in bone. So this is probably traces of dentin. And with quantities, a number of amino acids covered that is much lower than what associated with the material 2 million years old or more recent. So here we cover a bit less than 400 amino acids. Profile of peptide length distribution that is highly skewed toward small fragments, high level of deamidation. And measuring arginine to ornithine conversion, we see that it is particularly high in the Arctic Miocene rhino. So the possibilities and the information that we get from the sequence are associated with the possible structure and distribution of the various species of rhinos. And here we have preliminary recoveries that place one of the groups, and in particular the Alasmoterini, that's without going too much into detail, that's split with a certain distribution. Our results show that based on the amino acid substitution, our sample split before the others, and this can have a different explanation in terms of how rhinoceros evolved and arrived there. I'm going to method development, and I'm going to conclude very soon. There are 2 objectives. It's not only going for the most ancient sample, but also increasing the throughput, because the analysis of each one of these takes 2-3 hours in the instrument. But at the same time, there are cases where it's interesting to have characterization of large data sets. So small bone chips where you are interested in determining to which species they belong to, or sex determination of an extended set of samples. So to do this, in 22, this paper appeared on Nature Communication where we described SPIN. This was Patrick's main work. High level of automation at sample preparation using the Kingfisher and something similar to PAC, starting from 5 milligrams. We did a lot, Patrick did a lot of comparison between DDA and DIA. And this also led and inspired another approach for the automated high throughput sex identification of human remains starting from dental enamel, but in this case, without cutting a fragment of enamel. It's Claire's work, because we use acid etching. So the enamels got put in contact with a drop of acid. The drop of acid is immobilized on an evotip, analyzed on the Exploris with PRM workflow, and then processed with Skyline. And the attention is focused on the quantification of five peptides, 2 from female, 3 from male individuals, 2 from AMEL-X, 3 from AMEL-Y, sorry. And the purpose is to be able to process hundreds of samples per day. This is particularly interesting for archaeologists or people that wants to characterize entire population or demography, especially for subadults, so children or juvenile individuals. We tested it first on 10 materials from Jordan, 10,000 years old, so adverse conditions, and then on material from Europe, and in particular from Austria, that was of historical origin. And now we are trying on a Bronze Age material, so material that is 5,000 years old. I'm going to conclude promoting the training activity we do. We have a practical paleoproteomics summer school that runs second half of every August. Now we started 5 years ago in 2020. And it's 1 week of lab work, data generation. So the students generate their own data. They don't touch the mass spectrometer, but they generate their own data, and data analysis with MaxQuant. And there's 3 days before of lecture. And the lecture can be also chosen as a specific module on its own. So it can be attended remotely, 3 days where we cover all the topics of interest at the moment. So lectures, lab work, here it is, and the program for the first 2 days, for the first 3 days. So history of paleoproteomics, application to cultural heritage, and forensics and evolution. We're also hiring. This is the last slide. So if there's anyone interested in transitioning to ancient proteins, so taking a break for quick reaction and focusing on something old, we will open a position for a postdoc soon, focused on mass spec method development for paleoproteomics. And with this, I conclude, and I thank you all.